Hi, it's Rick Powell from Nanoprobes here, and today I'm introducing a new product, Click Nanogold. These are nanogold gold nanoparticle labels for bioorthogonal electron microscopy labeling. These are based on the original Click chemistry reaction. That's a cycloaddition reaction between an azide and an alkyne to give a triazole 1,4 isomer linking the two components together, and it's catalyzed by copper 1. What makes it click? Proceeds under physiological conditions. There's no cross-reactivity with endogenous components of tissues or cells. It's a fast and efficient reaction. The reagents and the products are both non-toxic, and that means it can be used in living cells and tissues without fear of cross-reactivity or reactivity with components that are present naturally. Click reagents are one of a class of labels called bioorthogonal labels, which means they take advantage of reactions that don't occur naturally in cells or in tissues or other biological systems. Other examples include the SNAP and CLIP tags from New England Biolabs, the Halo tag from Promega, or the FLAG tag, which is a peptide tag that can be engineered into target proteins and then recognized by antibody probes. The advantages of bioorthogonal labels is that they use conjugation reactions that don't occur in natural cells or tissues, so they can be used without cross-reactivity towards anything that's already present. They're non-toxic and biocompatible. They stay reactive during long experiments. That means that they can be used to tag components at specific points in dynamic processes as you monitor them, where you want to observe in detail the exact distribution of a target at a certain point in a process that you're studying. Here's the reaction that's used for our nanogold labeling reagents. As mentioned earlier, it's the cycloaddition reaction between an azide and an alkyne to form a triazole conjugate linking the two components together. And this is called the copper-1 catalyzed azide-alkyne cycloaddition, or CuAAC reaction. These are the products that we're introducing. The first is Mono-Azido Nanogold, which is a 1.4 nanometer nanogold particle functionalized with a single azido group. It's reactive towards alkynes. And this targets alkyne modified components, usually proteins, nucleic acids, or other biological molecules that have a synthetic alkyne incorporated into them. The other new reagent is Mono-Alkyne Nanogold, which is the complementary part of this reaction. It's our 1.4 nanometer nanogold particle functionalized with an alkyne group, which reacts with azides that have been incorporated into target proteins, nucleic acids, or other biological molecules. The features of these reagents include bioorthogonal labeling, no cross-reactivity and no reactivity with background or endogenous cell or tissue components. Mono-Azido Nanogold also works and reacts with strained alkynes or other unsaturated compounds that are used in some copper-free click reactions. So if copper is an issue for you, then use Mono-Azido Nanogold to label a strained unsaturated target. They're stable and biocompatible. They're soluble, but that solubility is incorporated without introducing any ionic charges. So there's no binding to charged proteins, nucleic acids, or other cell or tissue features. They're well tolerated by living cells. You can use nanogold reagents and conjugates for live cell imaging without disrupting cell viability. They're ultra high resolution. The click reaction is a very small chemical entity, and there's no need for targeting probes such as antibodies or proteins. These put the nanogold right on the target site, and you can use for high resolution electron microscope techniques to see targets at macromolecular resolution. They can also be used for optical visualization. With silver or gold enhancement, you can make the nanogold label visible for light microscopy. And on blots, you can use enhancement to see as little as a picogram of target IgG, making this a super sensitive detection technique. If you're doing ultra high resolution electron microscopic techniques with specialized methods, like single particle averaging, we also offer the smaller undeckergold reagents, which are only 0.8 nanometers in diameter, with the same reactive functionality. Applications of these products will include labeling components of vital processes in living cells, Again, ignore endogenous components like biotin or cross-reactive proteins. Monitor a dynamic process and tag just when you want to. High-resolution labeling of subunits or molecular sites. Just incorporate the corresponding reactive azide or alkyne. You don't need an antibody or other large targeting probe. You can multiplex your labeling by adding a click reaction to detect an additional target for which there isn't a suitable antibody or probe, or where cross-reactivity would be a problem with antibodies. So to summarize, Click Nanogold is a bioorthogonal label. It's stable and biocompatible. It's well tolerated by living cells. Provides ultra-high resolution electron microscopic labeling. Can be used for optical visualization or supersensitive detection. And if you're doing specialized high-resolution electron microscopic methods, the smaller undecagold reagents are available for even more precise labeling. Now, for using these reagents, the key is to plan your labeling. Usually, the separation method will determine what stoichiometry you use for reaction. When you're labeling with Click Nanogold, like with other nanogold reagents, you'd usually use an excess of the component that is most easily separated. We usually recommend gel filtration liquid chromatography as the preferred separation method. And that means that the component you use in excess will usually be the smaller one. So for molecules smaller than nanogold, such as peptides, you'd usually use an excess of the smaller molecule. And we usually recommend a 5-fold to 10-fold excess. If you're labeling a molecule that's larger than nanogold, like an antibody or a protein, you would use an excess of nanogold, between 2-fold and 5-fold. And we usually recommend starting with a 3 or 3.5-fold excess of nanogold. How do you know whether nanogold is larger or smaller? Well, it's about the same size as a protein of 8 to 10 kilodaltons. The nominal molecular weight of nanogold is actually 15,000, but because the majority of that comes from heavy gold atoms, the actual size is smaller than a protein of comparable molecular weight. To use Click Nanogold, there's a couple of factors to bear in mind. The copper-1 catalyst is generated in situ from copper-2, so in addition to the Click Nanogold, you'll also need to have present a source of copper, which in this case is copper-2 sulfate, and that's reduced to copper-1 during the reaction. You need a ligand to stabilize the copper-1 and allow it to catalyze the reaction. And in our case, this is tris-3-hydroxypropyl triazolyl methylamine, or THPTA, which coordinates to and stabilizes copper-1 ions. And finally, a reducing agent to reduce the copper-2 to copper-1 so that it can initiate the reaction. And that's sodium ascorbate. The procedure for using Click Nanogold is actually the same for both of these products, since it's essentially the same reaction. Now, for using the larger 30 nanomole packaging, you need to prepare the following solutions. 500 micromolar alkyne modified or azide modified molecule to be labeled in 0.1 molar phosphate at pH 7.0, usually sodium phosphate, with 10% dimethyl sulfoxide. And the dimethyl sulfoxide ensures that the nanogold remains dissolved and can undergo reaction and doesn't precipitate or aggregate. 15 millimolar THPTA in water, 6 millimolar copper sulfate, and 300 millimolar sodium ascorbate in water. As a first step, mix 10 microliters of the 6 millimolar copper-2 sulfate solution with 20 microliters of the 15 millimolar THPTA in a conical tube. And that'll be your catalyst for the reaction. Next, add 0.5 milliliters of deionized water to your 30 nanomoles of Mono-Azido or Mono-Alkyne Nanogold. Then vortex it. That'll give you 0.5 milliliters of 60 micromolar nanogold reagent in 0.1 molar phosphate buffer at pH 7, to the ideal conditions for undertaking this reaction. And that's the buffer from which these reagents are lyophilized. Next, add 60 microliters of your solution of the molecule to be labeled to the nanogold reagent solution, vortex to mix, and then add that mixture to the mixture of copper sulfate and THPTA. Finally, add 10 microliters of your sodium ascorbate solution to that mixture from step 5, vortex to mix, and then agitate gently, usually on a shaker at room temperature, for about an hour. If it's convenient, you can then refrigerate overnight and separate the next day. For the 6 nanomole sizes, the procedure is exactly the same, but you just use 1/5 of the quantities. The concentrations of the reagent should be the same. The procedure is the same, and the time lengths are also the same. And here's some references for these reactions. And you can go to these references to look up the details and find suggestions for modifying or optimizing them if they don't give you ideal results the first time you try them. You can download the slides from this presentation on our website, and we'll give you the URL for that later on in the presentation. Now, separating the conjugates, we usually recommend gel filtration as the preferred method for separating nanogold conjugates. And this separates larger nanogold conjugates from both unlabeled conjugate biomolecule and unreacted nanogold, as well as from larger multimers, precipitates, aggregates, or larger gold particles. As mentioned before, the strategy is to make the larger reagent the limiting reagent, so it reacts completely and there's none of it left unreacted. And that's usually because it's more difficult to separate the larger reagent from the conjugate. It's easier to separate the smaller reagent. Now, look for a size difference of at least a factor of 2 between the conjugate and the reagent that's used in excess. And for equipment, this usually works well with standard liquid chromatography. You don't need to go to high-performance liquid chromatography or advanced chromatography systems. To set up the separation, equilibrate the column for 2 column volumes before you inject the reaction mixture. Reduce the injection volume to be at most 5% of the column volume. And for best resolution, try and make it smaller. If the reaction volume is too large, then you can use a centrifuge filter, like the centricon filters from Millipore, to reduce the volume. And if you're labeling something larger than nanogold, the 30,000 molecular weight cutoff filters work well. If you're labeling something smaller, then the 10,000 molecular weight cutoff works quite well. And you can use these to reduce the volume before injection into the column if you need to, or if necessary, go to a larger column. When you're running the column, you should monitor at 280 nanometers if you're labeling a protein, or 260 nanometers wavelength for oligonucleotides. That will let you monitor the amount of protein or oligonucleotide that's eluting. Now, to monitor the nanogold, monitor at 420 nanometer wavelengths, which is where nanogold absorbs with a known extinction coefficient. You can monitor this visually. Nanogold and its components are both brown, and you will actually see them moving down the column. And here's an example of what you'll see. This is actually from a nanogold labeling of FAB prime fragments. And as you can see, Fab prime 2 and larger aggregates come out first, together with larger gold particles, followed by nanogold FAB prime, and finally unbound nanogold, both of which are brown, and then smaller molecules, which in this reaction have a kind of yellow color and follows a smaller peak close to the column volume. For selecting your gel, that'll depend on the size of the molecule you're labeling. If you're labeling antibodies or medium to large proteins, then Sucrose 12 or Superdex 200 from GE Healthcare, pre-packed columns or bulk media will work well. Now for Superdex 200, there are actually 4 different matrices to choose from, so pick the one which corresponds best to the range of the size of your molecule to be labeled to the nanogold. So those two, the molecule to be labeled and the nanogold, should really be towards the ends of the size separation range, and that'll give you the best resolution for separation. For smaller Fab prime fragments or other antibody fragments or smaller proteins, then Superdex 75 works well. If you're labeling larger proteins, then Sucrose 6 or even A1.5M from Biorad have an appropriate size for separating those. And if you're labeling very small proteins and peptides, then Superdex peptide pre-packed columns or Superdex PG30 bulk media from GE Healthcare is a good choice. For small molecules, then simple desalting gels will work quite well. on molecules 2,000 or 3,000 molecular weight and smaller. Now we've included the URLs at the bottom. As mentioned, you can download the slides for this presentation, which have active links to all of these pages, and we'll give you the address to download those slides later on in the presentation. You can also use spin and gravity columns, and here's some examples of the spin and gravity columns which are appropriate for separating nanogold labeled proteins and other nanogold labeled molecules. And here's a couple more spin and gravity columns, very different types of conjugate biomolecules. You can also use gel electrophoresis. However, you should use caution interpreting the gel shifts because they're often not good indicators of labeling. Although nanogold has a molecular weight of 15,000, the gel shift may be much smaller, and it may be smaller even than the apparent molecular weight of nanogold. The gel shift in some of the gel separations that we've done is about 2,000 to 4,000 molecular weight. Use non-reducing gels. Thiol-containing reagents like dithiothreatol can break down the nanogold particle, and so that can be a problem for if nanogold is exposed to these types of reagents for any length of time, and particularly over the length of time required to run a gel. To make sure that you can confirm labeling, you should detect each component separately. That means running 2 lanes or 2 gels. Stain one with silver or gold enhancement, which is the visual detection method used to detect nanogold, and stain the other with a protein stain like Kumasi blue. The nanogold conjugate will show up in both lanes. Note that silver or gold enhancement are highly sensitive detection methods. Monitor carefully visually while you're doing silver or gold enhancement, and stop when you see the bands. Otherwise, you may develop a tail behind the bands that obstructs resolution. And don't be confused about silver or gold enhancement and use a general protein silver stain. The silver stain to develop proteins is a different chemistry to silver enhancement. So make sure that you're using silver enhancement and not silver staining. Nanogold labeled oligonucleotides can be separated by reverse phase chromatography, which separates unlabeled oligonucleotide, unconjugated gold, and conjugates. For best results, we usually find a butyl C4 column works well, but in some cases biphenyl or C18 columns will also give good results. Elute with a gradient of 0 to 70% acetonitrile and 0.1 molar triethylammonium acetate buffer at pH 7. And if that doesn't give you sufficient separation, you can consider hydrophobic interaction chromatography, which often gives better resolution. Gel electrophoresis also works for nanogold labeled oligonucleotides. And again, to confirm labeling, divide the reaction mixture in 2 and run 2 gels or 2 lanes in parallel. Treat one with a standard DNA stain, like ethidium bromide, to visualize DNA-containing species, and visualize the second with silver enhancement or gold enhancement to visualize the nanogold. Compare the silver-enhanced, gold-enhanced, and ethidium bromide stained lanes or gels. The same band is stained in both treatments. That's the labeled oligonucleotide. How do you quantitate labeling? How do you know if it's worked? We recommend using the UV visible spectra of the conjugates and comparing that with the UV visible spectra of the unconjugated components. Here's the UV visible spectrum of nanogold from our website. This profile is reproducible and consistent. The extinction coefficients are known, and the ratio of the extinction coefficients at different wavelengths is always the same. Here's the UV visible spectrum for comparison of a nanogold labeled protein, in this case a Fab prime fragment. As you can see, the absorption at 280 nanometers is greater because the Fab prime and the nanogold both absorb at this wavelength. If you were to overlay the two spectra, you would find that the absorbance at 280 has a contribution from the nanogold and from the Fab prime. So to calculate the labeling, you would use the ratio of the nanogold extinction coefficients at 280 nanometers and 420 nanometers to calculate what fraction of the measured absorbance at 280 nanometers is due to the nanogold. Subtract that from the measured absorbance. The difference is the absorbance due to the Fab prime or the labeled conjugate biomolecule. Use that difference to calculate the concentration of the conjugate biomolecule using its extinction coefficient, and use the absorbance at 420 to calculate the concentration of nanogold using its extinction coefficient. The efficiency of labeling is the concentration of nanogold divided by the concentration of the conjugate biomolecule, and that'll tell you how many nanogold particles you have per conjugate biomolecule, which is how well the labeling has worked. For reference, here are the extinction coefficients again of our Nanogold and Nanogold gold nanoparticle labeling reagents at 280 and 420 nanometers. And for comparison, as an example, here are the corresponding extinction coefficients of Fab prime and IgG antibodies. Again, this is all available on our website, and in the presentation slides, you have a link to our website to go and check this and go step by step through the calculation procedure. Ordering information for these products is given here. All are packaged in 3 different packagings. The Nanogold reagents are available either as 1 vial of 30 nanomole for larger reactions, 5 vials each containing 6 nanomoles if you're doing multiple smaller reactions or wish to optimize a procedure over several reactions, or for introductory or smaller labeling reactions, 1 vial of 6 nanomole sizes. For our Nanogold reagents, they're available as 1 vial containing 50 nanomoles, 5 vials of 10 nanomoles, or a single vial of 10 nanomoles. In the future, we're going to look to develop Nanogold and Nanogold DBCO reagents for copper-free click labeling. So keep an eye on our website for new product announcements. They'll be announced there first, www.nanoprobes.com. Here is our website. Please visit to check out the instructions and detailed information about these or any other of our products. And here's our contact information. For sales or customer service or general questions, email nano.sales@nanoprobes.com. For technical support, it's tech@nanoprobes.com. And here's the URL for downloading this presentation from our website. Thank you for listening, and good luck with your labeling reactions. And we hope that our new Click Nanogold reagents are useful. Thanks again. This is Rick Powell at Nanoprobes. Have a good day.